Hello students, welcome to the SRBPS virtual classroom program. I am your biology educator Prerna Rathi. So here I am once again back with the concepts of cell cycle. In the last class we had discussed about mitosis and its significance. Now let us discuss what is meiosis and what is the significance of meiosis. Meiosis, my dear children, is a specialized kind of division which reduces the chromosome number by half. Hence it is known as reductional division. It results in the production of haploid daughter cells. It involves two cycles of cell division actually and nuclear division as well, meiosis 1 and meiosis 2. Single cycle of DNA replication, hence the DNA does not get to replicate itself the second time. Hence the total DNA will be divided into half in all the four daughter cells. Also, there is a pairing of homologous chromosome and there is a recombination that occurs during the meiosis 1. Let us see one by one how all this happens. The meiosis is divided into two phases, the meiosis 1 and meiosis 2, which are further divided into same respective phases like prophase 1, metaphase 1, anaphase 1 and telophase 1, whereas meiosis 2 consists of prophase 2, metaphase 2, anaphase 2 and telophase 2. Let us start with meiosis 1. Meiosis 1, students, is actually the reductional division. Let me explain briefly what happens. Just like the mitotic phase, in the prophase what will happen? The nuclear envelope will be dissolved, proper chromosomes will be formed. In the metaphase, they will align at the mitotic, align at the metaphasic or the equatorial plate. During the anaphase, they will move towards the spindles, the centrioles. Then again the nuclear envelope will develop during telophase, and after this two separate cells will be formed, both of which shall be diploid, but the division is reductional. Okay, so this will be like n n n n, two copies will be there so that they can form daughter cells of n each. Okay, so this is hence known as reductional division. Let us discuss the prophase 1, which is a very important phase of meiosis 1. It has been further divided into leptotene, zygotene, pachytene, diplotene and dikinesis. Let us discuss what happens in leptotene. During the leptotene, children, the chromatin material will start forming chromosomes. So condensation of the chromatin will begin. During the zygotene, whereas the chromosomes, the homologous chromosomes will start coming together. They will start pairing up, because after this they need to form a complex known as synaptonemal complex. There is one chromosome and the homologous chromosome is this one. So they come together to form a synaptonemal complex, which is visible in the form of a bivalent or a tetrad. If if not clear threads are not visible, then it is a bivalent. If clear four parts are visible chromatids, then it is a tetrad. I hope this is clear. The next stage is very important. It is called the pachytene. Proper bivalent now appear as proper tetrads, where you can see very clearly how each of the chromatid is clearly visible. During this phase, the crossing over will occur and there will be exchange of fragments between homologous chromosomes. Okay, after this, can you see this tetrad has joined each other in order to exchange its fragments. During the diplotene, the synaptonemal complex will segregate and there will be formation of a structure, an X-shaped structure called chiasmata. Fine. The other parts, these parts are going to move away slowly. This cross-shaped structure and the point where recombination is going to occur is also dissolve away. Fine. At the end, the terminalization of chiasmata will occur during dikinesis. Chromosomes are fully condensed, and after this it will enter the metaphase. Let us see what happens during the metaphase. Same thing, it will align itself at the metaphasic plate or the equatorial plate. Also the centrioles take their position, spindle fibers are joined to the kinetocores over the centromeres. Understand? Then during the anaphase, as you can see, the spindle fibers start shortening, thus pulling the chromosomes towards each other. They will move towards the pole. During telophase, the nuclear envelope will redevelop and then the cytokinesis will occur, where two cells will be formed. Those two cells, students, will enter prophase 2, metaphase 2, anaphase 2, telophase 2, and final cytokinesis, which leads to formation of 4 haploid cells. So in the beginning what we had, listen very, otherwise it is going to be, then it is going to be equational. Meiosis 2 is equational, meiosis 1 plus meiosis 2 together it is reductional in nature. Fine. So we've already discussed what happens during prophase 2, anaphase 2, telophase 2. Now, the significance of meiosis. Why is meiosis important? The conservation of specific chromosome number of each species. If we have to maintain the number of chromosome as 46 in human beings, then it is important that our gametes be 23 each. Otherwise, during fertilization, the chromosome number would become magnificently high, which we have to not allow. Also, a reduction of chromosome number by half can happen only by meiosis. Apart from that, all this evolution, the genetic variability that is caused is because of the recombination that occurs during the prophase 1 of meiosis 1. This recombination is the key to life. The change that is occurring is because of meiosis 1, prophase 1 of meiosis 1. I hope the concept of meiosis is clear to everybody. That's all for today's class.